starting our class. Welcome. And what we're doing this week is we're still talking about Chapter 9, and we're talking about rate limiting steps in transcription regulation, and we talked about the estrogen receptor, and there's lots more to say about that. And it's involved in chromatin remodeling, so that's why we're talking about it right now. And but we want to start reading this week Chapter 17. It turns out that Chapter 17 starts out with the nuclear receptors, because Chapter 17 is about receptors and signal transduction pathways. And we talked about estrogen receptor being a member of the nuclear receptors, of which there are 48 members in humans, including estrogen receptor and testosterone receptor and thyroid hormone receptor and progesterone receptor and glucocorticoid or corticosteroid receptor and vitamin D3 receptor and several more really significant receptors in our bodies for our health. And so we've been leading into Chapter 17, the whole course. The very first day we looked at FOXO as a signal transduction target like that. And so we'll do that. Then the flow for this is that section 4 that we're in goes like that. And our first exam is 2 weeks from Thursday, and it will include Chapter 17 and parts of Chapter 18. Chapter 18 is about cell cycle, and we've been talking about that a lot because that cell cycle is a target for a lot of this for normal regulation of our growth. For example, the estrogen receptor plays a role in growth during puberty in all female vertebrates like that. So it's good that we'll get to a piece of Chapter 18 because that's also been part of our kind of goal, is talking about that, because of course that's what gets disrupted in cancer like that, is uncontrolled growth. Yep. And so that's where we're at, and you're writing down questions and bringing them to class. Here's a question I got by email, and I just want to show this, and it starts and it says, I just had a quick question. So that's the first thing, is we are not going to criticize this student. It's a really good question. And it's a question about MIC and translocation, and they're just trying to clarify that, right? So here's something that comes up on the exams, and it's about wording and using words. And I'm not grading you on grammar. That's one reason they are short answers, right? Is it's not a big English course with essays, right? But the words that you'll pick on the exam, those can be really big stumbling blocks like that. And here's a good example, is that on the exam, sometimes a student will refer to the MIC cell, right? And that's not a cell. It's a protein. And that's just really different, right? So that is just a wording thing, and it's a thing where that'll come up on the exam. You'll go boom, boom, boom like that. You're explaining stuff, and you'll refer to one of these things, and it'll just be wrong, and I'll just be like, I'm trying to, gauge how well the student understands what's going on, right? And so again, it's not I'm not picky about grammar, but I have to be like realistic about the use of word choices like that. And so I just want to show this again, not a criticism to the student, it's just a chance to show something that hopefully will be memorable and helpful to everybody. So see, they say, I had a quick question. Going off the example of Burkitt's lymphoma, the antibody region of the B cell connects to the transcription unit of MYC and overall results in overexpression of MYC and B cells. Is that correct? And you kind of go, yeah, that's it. But we just think that from the the sort of the fundamentals, we'd want to be sort of more precise. We'd want to be more precise on the language. Again, not criticizing this student because you're all learning this and would write or explain this stuff like this. So I responded, and I tried to put in big letters just to emphasize what I was saying. It said, well, we learned this term, the cis regulatory region, or the regulatory region, right? One of the fundamental things that we want to demonstrate on the exam is that we understand that each gene has two parts, and we call one part the transcription unit, and we call the other part the regulatory region, or the cis regulatory region. So given a chance, you want to say that, right? It's the cis regulatory region of the antibody gene. Remember back on the first quiz, there was a question, what does MyoD protein bind to? And some, really good students just wrote enhancer, like that, the enhancer, or enhancers. And I'm like, yes. No, because every gene has an enhancer, but it's the myogenic gene enhancer, right? We just have, and again, I'm not like a jerk, like that, but just to demonstrate, that you understand the principles, it's going to be necessary to just go like that. And you don't need to be anxious about it. And if it's like, English is your second language, you don't be anxious about it because it's not grammar, really, and it's not structuring a big essay, but it's like getting the words correct, the word usage correct. So I would say in Burkitt's lymphoma, partly because it's Burkitt's lymphoma is always this. It's always a translocation. That's how they defined the disease, the cancer, Burkitt's lymphoma is. They could see back in the 1950s a specific translocation between two specific chromosomes. And so Burkitt person says, that's a certain kind of cancer. And they said, yes, we'll use that in diagnosis. So Burkitt's lymphoma is always this situation where there's been this translocation. And it's the cis-regulatory region, right? It's the cis-regulatory region of the antibody gene, right? And we don't say gene because an antibody is a protein, like that. So we want to say antibody gene, right, to make sure we say that. And, like, I'm really easygoing and I'm not really fussy and picky. It's just I want to show you this because it's just the way it has to be, like that. You really have to strive for that, not be anxious about it, but just be thoughtful about how you're saying, using the words, right? Like that. So, and the cis-regulatory region of gene connects to the transcription unit of MYC in B cells. And it's good to say that these B cells are what normally express high levels of these antibody genes. And so that's the problem, right? You're saying the problem, right? We got two parts of every gene, and there's part of a gene, the antibody gene, that is expressed at high levels. That regulatory region says, make lots of this, and that gets connected to MYC, and we just make too much MYC, like that. Another thing you can do is you can draw that out. And so lots of people are, well, I'm not an artist, but you'll be asked to draw some stuff on the exam. And you can also use a combination of words and drawings to really get your point across. Again, label them, simple label drawings, and just show here's an antibody gene, here's a MYC gene, and I'm connecting this part of this gene with that part of that gene, like that. And so, great question. I want to super encourage students to ask questions in class. And we do that concept chat partly for you to vocalize, start saying these things to get practice, right? Because learning is about repetition. Like learning a song is about, well, you got to recall it and repeat it like that. And so it's really good. In general, I don't, like when I was in college, I didn't study with a partner or study groups, but I think it is good in some courses to have somebody that you study with that you get a chance to say it or show it or draw it with another person and talk about this just to smooth out your ability to sort of express, what you're learning like that, okay? So we're getting ready for our exam. Our exam is 50 short answer questions, 2-point questions. What's this? What's that? Like that. I think it's a kind of flashcard style learning. Like, what is this? And you like that, Okay. So just that. I want to show you that. Then going to be putting signs up for these biomedical research internships. That is this kind of research in medicine, just the kind of stuff we're learning about that happens in Scarborough. It's just the most fantastic opportunity. It's better than any place I've ever been. It's just fantastic. It's so supportive. Eva's working there right now, and so you can talk to her about it. Hannah's working there right now. You can talk to her about it, and then other classmates that you've have right now are, because they have like 12 students down there, and that's for this coming school year, like that. Just an example. It has a summer program too that is any student can apply to from any school, and they just have hundreds and hundreds of applications. It's a really good opportunity. For the school year program, it's just us, so we place students down there. It's really good experience. Okay, then Riley, I want to always keep telling you about Riley. He's part of our course, like that, okay? All right, so this is where we're at, and so we're saying one way to drive through a big part of Chapter 9 is to add to our learning, right? We started with the clay model video. That's a big part of Chapter 9. And then how do these transcription factors do their job? And the answer is, well, each gene has a rate-limiting step, and that's what's regulated. And there are 3 sort of more famous rate-limiting steps. There can be others, but these are the big 3, like that. We talked about MyoD, and MyoD is this one that's really sweet because it gets us to name this protein that's part of the pre-initiation complex. It's TBP, TATA binding protein. And they call it that because some promoters, about 70% of promoters, have TATAA as part of the sequence for the promoter. And who binds there? TATA binding protein, because it looks like TATA letters, TATA, like that. So TATA binding protein, TBP. So we learned about that. That's really what myo does in myogenic. So see this thing we started our course with, right? There's myo-D right there. And here are the proteins of the promoter. In that video, I just made them all the same color. I made several. They're all the same color. Well, we could go back to that video, right, and make another one and be like, hey, look at that TBP binding to myo-D, helping myo-D bind to enhancer. And then it's got, a specific role to play there. So myo-D's role is to help get it there and help assemble this pre-initiation complex. So we're just adding to that answer. And there are lots of transcription factors that regulate some aspect of this pre-initiation complex just to help it get going. Yep?
So is TBP in this example a co-regulator, or is it bound to the DNA, and that's how it gets from D to the enhancer?
Yeah, so that's a good question because we have these transcription factors, and some of them bind directly to DNA, and so they must have a DNA binding domain, like we learned about myo-D. And so what about this TBP? TBP binds directly to the DNA. It binds directly to the TATA box. It just needs help. It needs help kind of, I'm a little bit lost, and it needs to kind of get tethered and help get it there. But it binds directly, so we would just call it a transcription factor.
Okay.
Its other name is TF2D, right? And because it's like they labeled up all of the transcription factors that bind to the promoter like that. And TF2D stands for transcription factor 2D. But TBP is a much funner name to remember because it's the TATA binding protein, like that. Yep? Yes, and then more generally back to that thing. So some transcription factors have DNA binding domains and bind directly to the DNA, and we just call those transcription factors, like myo-D and the estrogen receptor. Then some bind indirectly, and we call those cofactors or co-regulators, and they could be co-activators or co-repressors, and we met a couple of those last week when they help the estrogen receptor do its job, and we'll review that. Okay, so because that's the first one, we had myo-D, and that's a good one to pick, but there would be hundreds of transcription factors that fit into this category. They work by regulating the pre-initiation complex in one way or the other. If it's not TBP, it's some other pre-initiation complex protein, including RNA polymerase itself. We need to help get it there. Then we move down to this remodeling chromatin, and chromatin is the word for DNA plus proteins bound to it. When you see a picture of a chromosome, it's DNA plus a bunch of proteins bound to it. And remodeling chromatin just means that those proteins are folding the DNA up. The DNA needs to be compacted to fit in our cells, and it can be so compacted that the DNA is like folded away and inaccessible, and we need to open it up. And that's why we talked about the estrogen receptor, because the estrogen receptor and the nuclear receptors in general regulate transcription at a rate-limiting step that involves unfolding the chromatin. And I don't know, I guess I got that a little bit out of place, but that's fine. And so we could go back to this drawing, and I could just make another video about the estrogen receptor. And I talked about that this image is about muscle differentiation and myo-D, but we just can fill in the blanks, right? So two cells. In this case, what are the cells in the context? Well, like any vertebrate juvenile, like a human child, female child, is got the estrogen receptor there, and then puberty and adulthood, like that. So, we could just switch these labels around. What are you talking about? The DNA here, the clay would be estrogen response genes, of which there are many genes that the estrogen receptor activates in puberty, like that, okay? And then, what's the transcription factor? The estrogen receptor, like that. So we dive into that, and we don't show this here, but we couldn't show in a clay model that here, the DNA, the clay would just be all twisted up and unavailable. And then here, in puberty, it gets opened up, like that. Okay, so that image is still with us. We just plug it into a different context, okay? And then this is image 2 from that in-class drawing assignment, where these are genes, and this is the myo-D gene, and it's got to get transcribed. into mRNA, then translated by a ribosome into myo-D protein. Then it goes back in the nucleus and binds to the enhancer of the myogenin gene, activates its transcription, then the mRNA myogenin has to come out and get translated into a protein, and it's a transcription factor like that. Well, the same is true for any transcription factor. They're all proteins, and so they all need to be expressed. And so it's true that there is an estrogen receptor gene, and it needs to be activated like that, okay? And remember I said, you know, that as you go out here, there is sometimes that this is regulated. So you can think of these as dominoes falling, and you can think of the estrogen receptor as a very interesting domino, because here's a series of dominoes that lead to the activation of expression of the estrogen receptor, and it's expressed. And there's no estrogen during the years and years of childhood. So the dominoes just pause, waiting for the estrogen hormone. So it's a series of feedback loops that go out into the cell, and then can be dependent on a hormone or a ligand, a molecule regulation from some other part of the body, which in turn could be regulated by environmental factors like nutrients or seasonality, like that. And so we have this really static DNA that's going to get played out, it's going to get expressed, it's going to be output, and part of the output story are these transcription factors, which in turn are coded for by the DNA. And so the DNA sort of stores a kind of symphonic score where these things are expressed, and then they can be regulated. So we have this progression of expression and playing out of the DNA information. Okay? So these are all good examples of that. So we're talking about the estrogen receptor, and here we are in chapter 17, and here it's just showing what we drew last time. So there is a nuclear receptor, and in the absence of the hormone, estrogen in our case, it binds this enzyme that we call histone deacetylase. Right? There's a longer name for it like that, but we like to call it histone deacetylase. And it removes acetyl groups from these purpley histones, and as a result, the chromatin is folded away. It's called heterochromatin, and it's not accessible for transcription. And these would be estrogen response genes. And then the hormone's added, like estrogen, and we switch the co-regulator from the co-repressor, histone deacetylase, to the co-activator. And both of those are enzymes, and they have the exact opposite catalytic activity. And so now in puberty, we start binding acetyl groups to, covalently binding acetyl groups to the histones, and they fold open like that. And the genes get expressed. So chapter 17 shows what we talked about last time, and it is how these nuclear receptors work. The nuclear receptors are ligand-dependent transcription factors. Okay? So that was how we got into chromatin. Let's talk about a transcription factor that regulates chromatin. Oh, estrogen receptor. That's our favorite. There are a lot that do with that, but estrogen receptor and the nuclear receptors are freaking cool transcription factors, because they have the three domains. They have a DNA binding domain that binds to the estrogen response element. It's an enhancer. Remember, they like to call it a response element because it both represses and activates estrogen response genes. So the estrogen receptor has that DNA binding domain. Then it has a trans-acting domain that's very interesting, because all it does is it binds either histone deacetylase in the absence of hormone or histone acetylase in the presence of hormone, like that. And then the estrogen receptor needs to have a third domain. It has to have the hormone or ligand binding domain, and they all fit together to do that really interesting switch function. Okay, so in chapter 9, the book really dives into chromatin and talks a lot about it. It's kind of a blizzard of information about chromatin, and so we need something to have to carry us through there. And our favorite thing is histone acetylation. And so this just shows DNA, and here these blue balls are called nucleosomes, and they're made up of histone proteins. And the book talks about that, and I have some figures about that too. So now we're talking through the chapter 9 a little bit in general about the chromatin. And the most sort of fundamental story of chromatin is, oh, the DNA is too long, and it needs to get wrapped up and folded up and shortened to fit inside the cell. And the first level of that folding is to wrap around these histone cores, these nucleosomes. And so this is what that looks like. And you don't need to know the names of the histones, but it's 8 histone proteins that are in the core of the nucleosome, and then it's one histone that helps kind of put it all together, like that. And it's these histones that get acetylated. And so you can look on Wikipedia too. I mean, I always think Wikipedia is easier to read than any biology textbook, which is sad but true, I think. So it's good to look at that. So in this context of chromatin remodeling, we will, in our course, emphasize 3 things. And one is DNA methylation. And we'll try to talk about these in separate settings. So we've talked a little bit about DNA methylation, and that's where a methyl group is covalently added to specific cytosine Cs. So in the DNA code, we have AGCT, and the C could be methylated or not. And when the C is methylated, it represses transcription. So we learned about that in the methyl videos, the differentiation. When cells differentiate, they methylate a lot of genes that we're going to use again. And people fit that story into chromatin remodeling, like that. And people like to talk about it a lot nowadays because there are a lot of different situations where the DNA methylating can occur. And when it does, it's in general repressing gene expression. There are different environmental factors that can really affect DNA methylation. Just one example would be if a child is systematically, like continuously bullied, they have changes in their DNA methylation pattern in their DNA that will last for the rest of their life. Like that. It's really hard to change some things that happen due to environmental factors in a person's life. And scientists learned methods to analyze the pattern of DNA methylation just in the last 15 years. And so there's lots of stories about it now because they're learning a lot about it. For us, when we think of DNA methylation, we especially think of differentiation. That's our favorite. It makes a lot of sense to us that, well, those genes aren't going to be used, so let's methylate them. And if we want to turn those cells into a stem cell somehow, we're going to have to remove those methyl groups for sure. So that's one. And we've talked about that some, and we'll talk about it more. The book talks about it some in chapter 9. It's a really different thing, so we want to kind of separate these stories. What we're talking about right now is histone acetylation. And that's what the nuclear receptors like estrogen receptor do to fat transcription, and that's the rate limiting step in the target response gene for those guys. Okay? In general, histone acetylation increases gene expression. The book talks about a lot of different ways of modifying histones. Unfortunately, one is methylation. So histone methylation is a type of chromatin remodeling, and that's confusing because it's not DNA methylation, it's histone methylation. The histones can get acetylated, they can get methylated, they can be ubiquitinated, they can be samoylated, like that, and the book is just like blah, 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 right? One way to remember what's going on is it's just going to affect transcription. It's going to affect gene expression. And in some cases, it's going to help increase it, some cases decreases. And it's about gene expression and transcription. And just how folded up is the chromatin? Is it folded away into closed heterochromatin? Or is it unfolded into open euchromatin? And so in talking through chromatin remodeling, yeah, there's DNA methylation, and then there's this histone modification, and we really like to talk about histone acetylation. That'll help us, guide us through that chapter, and you can add more to your knowledge, but you don't need to know about histone or simulation in this course, right? You just end up confused and throwing up outside. So, yeah, so we got to be realistic. You want to get a core understanding that you can talk about on the exam, and then add to that in the textbook. So in that, we have these terms, right? Open chromatin, euchromatin allows gene expression, transcription, closed chromatin, heterochromatin, repressive gene expression. And that's often the rate limiting step. So that's often what a transcription factor will regulate. Then there's a third chromatin remodeling we're going to want to talk about just briefly, and it's called nucleosome phasing, and we'll learn about that when we get to it. So those are our three favorite things to talk about with chromatin remodeling. That gets us a really solid start and a really solid way to drive through Chapter 9. DNA methylation, we'll talk about that at different points. It's DNA methylation. And then histone modification, we love histone acetylation. And then nucleosome phasing, we'll talk about that briefly. Yep, and that's just, I don't know why I put that in, just those, the terms like that, okay? And so in Chapter 9, the book shows this, and my gosh, right, it shows all the chemical details of that, and you don't need to dive into this in more detail. This is just showing the sequence of a histone, and there are specific amino acids that can get acetylated. In general, people like to draw this, that the histones form like a ball-like core, and as part of their amino acid sequence, they have tails, and the tails stick out, and the tails tend to be what gets modified, whether it's acetylation or any of these other forms. Those are getting modified. So that's what they're drawing there, and they actually call those histone tails, like that. And this is just showing that, and just showing the chemical reactions like that of acetylation and deacetylation, right? It's a chemical reaction with an enzyme, like that is the co-regulator, the co-repressor, co-activator, that is stimulating that. One thing then, what we've learned about in our course that's satisfying to think about is we learned that the enhancer, the response element where the estrogen receptor binds, that could be far away from the promoter and the transcription unit. But we learned that the DNA sequence is like a leash, and it's tethering that transcription factor to the area, like a dog on a leash. And so it is satisfying to think about that, because the estrogen receptor binds one of these enzymes, and that enzyme is now just concentrated in the region of that gene. And so it can just be bouncing along, either removing acetyl groups and folding up a big region of DNA, a big region of the chromosome. Or it can be adding acetyl groups and unfolding it, because it's just tethering the enzyme activity to this area. The enzyme was floating around like a dog running around your neighborhood. And then you put it on a leash, and now there's going to be a lot of biting on that area where the dog or the enzyme is located. So it's really satisfying to see that that's the way these transcription vectors work, is they're tethered. They're cis-regulatory elements. Cis means to leash them. We're just going to leash them onto this transcription unit, so they'll just be active here. MyoD binds TBP and increases TBP concentration, so TBP gets help finding the promoter. Estrogen receptor tethers these enzymes and just gets them at a high concentration right around there, and they're just modifying those nucleosomes and the histone tails on those histone proteins in the nucleosome, like that, okay? And again, you don't need the details of the chemical reaction. The book is showing that. It's great to see that. And certainly don't need to know which amino acids are that's being added to, like that. There's that figure again, and and our favorite is going to be that histone acetylation and deacetylation, and the estrogen receptors are a good example. Okay, I talked briefly about this. I just want to show you this, that in humans that an adult can get injections of testosterone, and that could cause any human to develop male secondary sex characteristics, or could get injections of estrogen, and that would cause any adult human to develop secondary sex characteristics of females, right? And that's because this role of estrogen receptor is occurring in both males and females. It's occurring in all of us and all vertebrates. And in the absence of estrogen receptor, in the absence of estrogen hormone, in juvenile females or in males of any age, is repressing. And that's what allows injections of the estrogen hormone to have an effect on a male. And, it could be biologically, the mechanism would be different, that a male could get a shot of estrogen and nothing would happen, right? It's testosterone that they're using to deliver secondary sex characteristics. But in our bodies, it works like that. And when we look at the molecular mechanisms of the estrogen receptor, and the testosterone receptor works the same way, it makes sense. In the absence of hormone, the receptor is a repressor. It's keeping those genes off. And then the hormone switches them on. And molecular biologists love that, because molecular biologists love the idea of a little molecular switches. Switch, switch, switch. It's just the prettiest thing. Have a molecular mechanism to say, oh, those genes are off. Oh, those genes got turned on. They really got switched on. And it's just such a satisfying explanation of how they got turned on. They get turned on and off by enzymes. Enzymes catalyze, the same reaction over and over again. So we just catalyze a whole bunch of changes to the chromatin. It really unfolds over this whole region, and it takes several minutes for it to happen, like that, because it's like that, right? So that happens. Then here's another thing, and this is something we'll want to tune in on a little bit, because this comes up in our section 4 about cancer. So there's a big-time cancer drug, and it's called tamoxifen. It's got different names. That's the one problem with pharmaceutical medicines, is they have different names depending on the company that's selling them and stuff. But the classic name for this is tamoxifen. And tamoxifen is an antagonist of estrogen. And an antagonist it inhibits. So let me just show you these terms, because these are terms from endocrinology. So the first is, we're using this term ligand. And that's a kind of a loose term, and it means a small molecule that binds to a protein and regulates it. So all hormones would be examples of ligands, but there are more things that are ligands. And we use that term, just hurrying along, we say, oh, it's a ligand, like that. Okay? This ligand can either activate or inhibit. And if a ligand activates a protein receptor, it's called an agonist, which is probably a new word for you. If it inhibits, it's called an antagonist, which is probably not a new word, right? An antagonist is sort of like your enemy. They antagonize you. And it's just interesting to see that the root word agonist, it means it's an activator. And these in endocrinology are really official terms. An antagonist and an agonist have to be known to bind to the receptor directly. Sometimes there are molecules that they don't bind to the receptor directly, but they have the same effect, and those are referred to as mimics, like that. So just some terms, and we're just using them in this context because you'll see we want to talk about tamoxifen. So when we say, oh, tamoxifen is an antagonist, it's an antagonist of the estrogen, that means it must bind directly to the estrogen receptor because it's an antagonist. We didn't call it a mimic. We said, oh, it's an antagonist. It binds directly and it represses. And so just to show you, there is the tamoxifen binding to that ligand binding domain of the estrogen receptor. All right, okay? And so tamoxifen is used as a chemotherapy in breast cancer. in our normal bodies, during puberty, the estrogen receptor stimulates growth of breast cells in females. And in some breast cancers, that growth control pathway is what's broken, mutated. And so the breast cancer is growing, and it's growing in what they say an estrogen-dependent way. Okay? So if a person ever has a biopsy, right? So they have a lump or a bump in their breast, and they have a biopsy, the doctors look at several things, and one thing they look at is they look at whether the cells have the estrogen receptor. And if they detect the estrogen receptor, the cancer is said to be an ER-positive breast cancer. And about 70% of breast cancers are ER-positive. That means they have the estrogen receptor. And then 30% are ER-negative. That breast cancer is out of control, growing out of control, but through some other problem. So this is just, and this comes up in chapter 20 when we're really talking about cancer biology, but this is our first kind of pharmaceutical example, I think, and that is doctors give tamoxifen to patients that have ER-positive breast cancer. It would just be a waste of time to give it to somebody who was ER-negative. And that should make just great common sense. The tamoxifen binds to the estrogen receptor. If the patient has an ER-positive breast cancer, they have the estrogen receptor, and it's helping, there's a problem, and it's causing part of the cancer growth. And we could stop that with tamoxifen. If the breast cancer is ER-negative, it would just be useless, right? And that happens, right, just out in the real world, right? Imagine somebody living on a street and they get breast cancer and it's ER-positive and they get tamoxifen and they go into remission. It doesn't cure it. It buys them time. And I'll give you an example of that. Well, I guess I can't, but because it's my sister. I'll try. So a few years ago, my sister in Minnesota got breast cancer, and it was ER-positive. And so they gave her tamoxifen, and they didn't have to hurry, so they did this. They then did surgery, removing the tumor. And they tried to just remove the minimum amount of tissue. And so they do that, and then when they're done with that surgery, they get some of the cells that still remain there, right? They removed a tumor, and then here are this neighboring cells beside that, and they call that the margin. And after the surgery, they check the cells in the margin to see if there are any cancer cells still there. Did we get all the tumor or not? And so they do it, and they say, the term they just say is, the margins were clear, right? So you're all set, right? So they did that, and the margins were not clear. So they gave a second surgery to my sister. They removed a little bit more tissue, and they checked the margins. After the second surgery, margins were not clear. They did a third surgery, and the margins were clear. And this was several years ago, right? So good, right? Seems to be everything's fine, right? How did they have the time, to just go slow and, move a little tissue, and then check and then have a second surgery? How they have the time? Well, she was taking tamoxifen, right? And it's not curing it, but it's definitely putting the cancer in remission. So it gives the doctors time to say, let's just go at this carefully and, we'll get it done like that. See? Like that. So imagine that somebody on the street has breast cancer and they get the tamoxifen and they get better, right? And then my sister's neighbor across the street gets breast cancer, and they're like, what happened? You're cured. What happened? How are you doing? And like, well, I had this tamoxifen. And that patient across the street goes to the doctor and says, my neighbor, she had tamoxifen. I really want to get that. And the doctor's like, no, yours is ER-negative. And they're like, it worked for my neighbor, just please, right? It's like that. That's what, pharmaceutical chemistry is like for cancer biology, is there are some treatments that are very specific, very targeted, and we'll learn about some of those in chapter 20. And they would work for this, right? It'd be useless to give tamoxifen to somebody with skin cancer, right? Or ER-negative breast cancer, like that. So that's a big take-home message, is that more and more of chemotherapy is very specifically targeted, and it works on that in a molecular medicine that we understand. So tamoxifen, antagonist of estrogen. So we're going to put that in, and this just shows, just a visual, right? That tamoxifen binds, so the estrogen can't. Like that cannot activate those genes, and those genes would stimulate growth of the cells, like that. Okay. And here's a Wikipedia, because it's been around. I don't know, it's been around for quite a few years, and it's very famous because it's very effective, again, at causing remission. And see, it's sold in all these different brand names, so you might not have heard of tamoxifen, even if you're taking it, right, like that. So just something we know about. And it says it was approved in 1998, right? So it's been around for quite a while, treated millions of people with hormone receptor positive breast cancer, like that.
Is it an injection, or is it like?
I don't know. Yeah, how do you take a pill or injection? I'm not sure. Okay, so that's one example, and they talked about that a little bit in chapter 20, but there'll be more talk of that, more talk of those kind of examples where you're like, okay, that makes sense. And I just want to bring it up now because we're learning about estrogen and it would make sense, right? So what does tamoxifen do? It just blocks the estrogen receptor from unfolding the chromatin. The chromatin stays folded up, the estrogen response genes stay off, and some of those response genes are genes that would stimulate the cell cycle. We're learning about chapter 18. Okay. So then here's another topic we want to talk about in chapter when we talk about the estrogen receptor, because it's just so relevant, right? So tamoxifen, big time antagonist of the estrogen receptor. Now we're going to talk just really briefly, just to kind of put this in some context. Now we're going to talk about an agonist, so something that could replace estrogen, and it just activates the receptor, right? That's what does it do? It just binds to the receptor, ligand binding domain, and it activates the receptor and unfolds those the chromatin for those estrogen response genes, and that's the rate limiting step, just turns them all on, right? So here's an example of a famous agonist, and it's this BPA that I don't want to drink a plastic with BPA in it. BPA is an estrogen agonist, and literally it binds to the estrogen receptor ligand binding domain, and it replaces estrogen. Who could that affect? Well, it can affect males and juvenile females, because they have the estrogen receptor and there is little or no estrogen around, but this BPA replaces it, like that. So that's who it could affect. And they are finding, fish, because it doesn't just affect people, they're finding fish in streams that are kind of both male and female, and they're typically sterile, but they have reproductive qualities and secondary sex characteristics of both a male and a female fish. Because fish and all vertebrates have the same sex differentiation strategy as humans, like that. It's not just BPA, there's just a whole bunch of chemicals that mimic hormones, and they're called endocrine disruptors, and they can cause really big trouble, like that. So I just want to mention that because so that's a kind of a really common thing in the current events and news, is these endocrine disruptors, and that's how they work.
So you said that endocrine disruptors mimic. Is that also a mimic?
So that's a really good thing, is that terms from endocrinology, right? So mimic means does the same thing. We don't know for sure, in certain cases, if that endocrine disruptor does that by binding to the receptor. Like just for example, a mimic could stimulate estrogen synthesis, right? And but in this case, we know BPA is an agonist, that it binds directly to the receptor. Yep. All right. Great. So just, this course could just easily be about the estrogen receptor. It's just, there's just so much, but we're driving through with that central piece of chromatin remodeling. That's how it works. That's how it functions to regulate estrogen response genes. All right? Great. Now we want to just talk a little bit about these nuclear receptors, because the estrogen receptors are an example of several. And the book talks about them in chapter 17, and we will try to streamline that. But it's just a gigantic, gigantic story in our health. So this is just a drawing of a figure of a nuclear receptor, like the estrogen receptor. They all have the same structure. DNA binding domain, transacting domain, and ligand binding domain, and transacting domain. Those three things, right? This is the part that would bind the histocellulase or deacetylase enzyme, like that. They all have that same structure. And they have a distinct way of binding to the DNA, like that. And so, just to say that DNA binding domain has zinc in it, so it was really easy for scientists to find that, because the zinc atom is coordinated by cysteines in the amino acid sequence. No big deal, but in particular, and the book you'll see likes to talk about this, is that the type of DNA binding domain is called a zinc finger domain, and all the nuclear receptors have zinc finger domains, like that. Okay, and then you can take this three-dimensional image, you can actually just lay this out as the transcription unit, the coding region of the estrogen receptor gene, and it's just laid out like that. So they have a really similar sort of format for all of these nuclear receptors, where you go from the transacting domain to the DNA binding domain to the ligand binding domain. Just another image of that. Just what it's showing here is just the amino acid sequence of any nuclear receptor. They all have a really similar sequence because they have really similar structures. And in our bodies, there are 48 numbers. So there would be 48 genes that code for nuclear receptors, and one of them is the estrogen receptor, one of them is the testosterone receptor, like that. And you don't need to scroll to write this down, but this just lists a bunch of them, and again, it's a super famous set of proteins in our bodies for our normal health. And this just shows a phylogenetic tree of the genes, because they're all related. And so this is a really good setting to learn about this concept, is the 48 members of this gene family are all related to a single common ancestral gene. So we have an evolutionary tree, not of different species, but an evolutionary tree of a family of genes that are all found in us. Okay? And there's a word for that, and that is paralog. The 48 nuclear receptors are paralogs, and we have a very definite, crisp definition of a paralog. Paralogs are two genes in the same species that are understood to share a common ancestral gene. Like that. And that's a new term that we want to, where it is. We already learned this term ortholog, two genes in different species. So for example, the estrogen receptor in humans and the estrogen receptor in cats are orthologs. The estrogen receptor in humans and the testosterone receptor in humans are paralogs. Yep. So the estrogen receptor in a human, the estrogen receptor in a cat, those are orthologs. And we love to talk about those because that allows us to study model organisms, right? We've got myogenin in us. We talked about myogenin in a dog, myogenin in a fruit fly. Those are all orthologs. So two genes in different species that are understood to share a common ancestral gene because the organisms shared a common ancestral organism. like that. You know, we're related to dogs. There used to be a common ancestor to both of us, and that common ancestor had myogenic, or the estrogen receptor, right? That's how we get an ortholog. Paralogs are within one species, where we talk about the paralogs and the different nuclear receptors within humans. So like the estrogen receptor in humans and the testosterone receptor in humans are paralogs. And they arise by gene duplication. So just an accidental duplication of a gene, and now we have two of them. And they can evolve to have somewhat different structures. This is just an example, like, you know, you had a screwdriver, and it got duplicated, and now you have two screwdrivers, and one of them retains the same ancestral function, and the other is free to evolve to have a somewhat different function. So we could have the estrogen receptor or something like that. It got duplicated, and it evolved and continued to be the estrogen receptor, and then we got another receptor doing somewhat slightly different. That's why all the nuclear receptors look so similar, is because they share a common ancestral gene. They have really similar sequences. This just to show you that these paralogs can diverge over time. They can be really different. Like, you can have a screwdriver evolve out a shovel into a shovel. That seems really different than a screwdriver. So you can have paralogs that have really similar roles, or they can start to diverge and be pretty different. And there are lots of these gene families, and gene families are sets of paralogs that arose by gene duplication and diverge to do somewhat different jobs, sometimes quite different jobs. The nuclear receptors are a really great example of a gene family in our bodies, and they're, you know, how they have somewhat different roles. Great. This is just an image that we've talked about. Here it's just the estrogen receptor doing its job like that. No big deal. Then this just shows some of the different ligands for the nuclear receptors, and you don't need to see the structure of these, but there's some really different ligands, and that's the key thing of the differences between the nuclear receptors. And just shows a different kind of evolutionary tree showing what some of the different roles are like that. And you know, just visuals, just to give you the feeling for that these are all related but different. They have some different stories, and they're fascinating because of their roles in human health, like that. Okay, great. All right. So, you know, as we want to talk about chromatin, we want to talk about an example. Well, the estrogen receptor is such a sweet example of chromatin and chromatin remodeling by catalytically acetylating or deacetylating histones in nucleosome. So, and when we bring that up, we're like, oh my gosh, you know, we're going to talk about estrogen receptor for a little bit because it's a member of a gene family of paralogs that are just really significant. And our book is talking about that at the start of chapter 17, because chapter 17 is about receptors and receptor signaling, like that. So there's some of that in there. And we're doing great. I think this would probably be a good time to stop and have a little concept chat like that. And a good time for me to get the sign-up sheet together. So let us talk about 3 things that you thought were important so far today. And so try to sit with just 2 people. So that's one thing. Right now there's a tendency to kind of group into larger groups, but it's good to have 2 people so people get a chance to talk. Remember, because I started the class saying, you know, it's good to get experience expressing your understanding, and it's good to have somebody listening to it, going, yeah, I don't think you have it quite right. I think it's a gene. So try to get into groups of 2. There could be one group of 3 if we have an uneven number. But we should, if you have 2 groups of 3, you have to break up into split like that. Right? So I see 2, 2, 2, and then see, and we have like 3 groups of 3 right now. So Eva's doing a good job breaking up one group of 3, and then yeah, we're good. Groups of 2 and one group of 3. All right, good job, everybody.
as a DNA test, so basically, last week's class, and we also added.
It has a what? Okay, so DNA I was like, wait, what is that? So it's combined to that receptor as well as DNA.
Well, I have DPA as
Well, I basically DPA was combined.
Oh, we'll do this for another minute, and think of 3 things that you thought were important. Oh, yeah. Okay, let us see what Ellie thought. Ellie, is what one thing that you guys are talking about you thought was important?
Okay, one of the things that we took away was the tamoxifen being like an antagonist of the ligand that prevents estrogen to binding at the estrogen receptor.
Yep. So, these nuclear receptors are ligand binding receptors that are transcription factors, and the estrogen receptor is a transcription factor that regulates transcription of estrogen response genes, and it does so in an estrogen-dependent way. And so in an ER-positive breast cancer, the estrogen receptor is expressed. So that's what it means. They can detect the estrogen receptor. And what are they detecting? Like in that, they do the biopsy, and what are they doing? And they, for the students in the lab class, they're actually doing PCR to see if they can detect estrogen receptor mRNA. Because if the gene's expressed in the tumor, there'll be mRNA for the estrogen receptor.
When you say gene expressed, is it like the estrogen receptor gene?
Yeah. Yeah, the estrogen receptor gene. So the DNA is always there, right? That's our message. The DNA, the genes are always there. When they get expressed, they're going to get transcribed into RNA. And of course, then for the estrogen receptor, we're going to make a protein. Scientists have come up with really easy ways of assaying the presence of the RNA. And then it's just implied, well, if it has the estrogen receptor RNA, it would have the estrogen receptor protein. But the easy thing to detect is the receptor RNA. So they just do PCR on these patients. And they biopsy tissue, they get a little of that, they purify the RNA, they make cDNA, and they do PCR. And they either detect it, and then it's ER positive, estrogen receptor positive, like that. And if they have that, then they must have the protein, and they find that tamoxifen is an antagonist. That means it binds directly to the receptor. It's not a mimic. It works by binding to the receptor, like that. And so that's a really good one, and it's just a really good example in so many different ways of helping understand what the heck is going on with some of the modern chemotherapeutic agents. They're specific, right? They're like, this is going to work on that specific kind of cancer, and it's not going to work on others. So older therapies like radiation therapy would be more general, like that. And now they have some very targeted, and more all the time, very targeted therapies like that. And they'll work on that, they'll work on that and just nothing else. So that's what that is, great. And then Godwin, what's one thing you guys talked about?
Oh, something about the parallel thing, and you have a question?
Yep.
One question that I was gonna ask with the parallel was like you were saying it's two genes in the same two genes in the species that share a common ancestor gene. I don't know if this I think I'm gonna be incorrect in what I'm about to say, but I was just like how you were talking about like ER positive breast cancer and how like the cancer cells contain an estrogen receptor. I was just curious of like the tie-in between like women that have like breast cancer and then like later in their life they usually develop like uterine cancer or something of the sorts. Is that like a parallel because they're both being like descended from like the same like they're both like estrogen dominant like cancers, or is that am I like way off?
I'm going to say you're way off, but I don't know, the idea, like I'm not, in any way a medical doctor like that, so I don't know, like this idea that, when a person gets one type of cancer and then they may later get a different kind of cancer, so I don't know about that.
Okay.
I know about the definition of paralog, and that's just focused on naming the genes.
Okay.
Let's just follow up with that, so, because it's, remember we now have the term ortholog, and we're using that when we want to talk across species. So, it's an ortholog when we're talking about a model organism. We're using mice, we're using fruit flies, we're using a different species to help understand our health. And it's because that species has an ortholog. It's like the same gene, function like that. And then paralog comes up, and especially in like more complicated organisms. So you really see very common that some simpler organisms have maybe one gene, and then a more complicated organism has paralogs of that gene. And so it's a little, the analogy of like with screwdrivers and like how so a lot of people just have a home tool kit with just one screwdriver. And then these people drive around with trucks that have tool kits across the back of the truck that has 40 screwdrivers, right? They're all screwdrivers, they just differ a little bit like that. And it's, it is a kind of a generalization, but that in creating more complex animals like a human, there tended to be a real expansion of gene families. And so you'll see that. You'll see that in a simple organism, there'll just be one or two family members, and then there'll be like 20 or 48, like that. Yep. So that's that. Then let me just say that, so we have ortholog, very definite definition of that. Then we have paralog. Then we have this other looser term, homolog. And homolog just means really similar. And we'll just describe this for a minute so you can see why there are a lot of scientists that just want to say homolog, right? Orthologs are homologs, paralogs are homologs, it just means similar. And then paralog gives a stronger claim, and ortholog gives a stronger claim. So here you have a situation, right? So you say, okay, the estrogen receptor in humans is an ortholog of the estrogen receptor in cats. And the estrogen receptor in humans is a paralog of the testosterone receptor in cats. And then you say, well, now how do you know that it's the estrogen receptor in humans that's an ortholog with the cat estrogen receptor, but not the testosterone receptor, because the human estrogen receptor is very similar to the cat testosterone receptor. So when you start talking about gene families and you start comparing the gene families in two species, then it gets a little complicated sometimes in explaining which one of those are orthologs. In this example, it's not too hard because you say, well, we have to look at the function, right? And the function of the estrogen receptor in both humans and cats is female development. And the testosterone receptor in humans and cats is male development. So you can't just go by the sequence. You have to look at the function in the organism to really get certain, more confident. But it does get frustrating when you have gene families between two species and you want to really try to sort out who's who. It takes some time. And so that's why a lot of scientists, will use the term homolog, and we see that a lot in that. And it just means let's take it easy. You know, we want to say they're really similar, and maybe we don't know which is which, or we just want to, you know, we want to just take it easy on that. So we have that homolog term, but ortholog is a great term, you know, because it comes with a lot of student understanding, and paralog is a great term because it comes with a lot of student understanding like that. So that's a good one. And Max, what is one thing that your group talked about?
We talked a lot about like checks and balances. So we talked about chromatin, like how it can either be open or closed, and how that's regulated. Since your genes are always there, right? There has to be some way to either express it or repress it. So we talked about that. With histones getting acetylated, or we talked a little bit about the estrogen receptor, and how if it was present or not, how you could use
Yeah.
tamoxifen.
Yeah. So checks and balances are, multiple steps of regulation. Yeah, that's a good concept too. Good broad concept that we're dealing with just about every day in our course. Yeah. Okay, great. And so we have more time, and people have questions about anything at all. Yep. Yeah.
Clarify a little bit about the histone methylation. That what you said, you said histone methylation. Could you describe that again?
Yeah. So let me find a slide to show that. And so we have this thing that we want to be careful about, and in going into this remodeling of chromatin is there are lots of different ways that chromatin gets modified. And we're going to have 3 in our course that we're going to be solid on. And one of them is DNA methylation, and we've already talked about that some right from the beginning of our course. And we'll keep plugging that in. And I mentioned bullying, and you're like, what bullying? Like that, right? Like that. There's a lot of different ways that the environment can specifically affect DNA methylation patterns, like that. Our favorite for our course is this differentiation, like that, like that. So, and we want to keep this separate from the other examples because it involves directly, covalently modifying the cytosines in the DNA molecule. Then there's a lot of changes that can happen to these histones. The histones are folding the chromosome, folding the DNA up, helping it condense, compact like that. And there are a lot of different modifications. Our favorite one is this acetylation. That's how these nuclear receptors like this receptor work. So we're really solid on that example. And I just said that the book lists, there are several different ways to modify histones. And I mentioned, well, one of them is histone methylation. And that just gets immediately confusing because it's not DNA methylation. And here's one thing to say about it is, just to mention it, histone methylation is just nothing we want to go into because sometimes histone methylation increases gene expression and sometimes it decreases it. And so that's just frustrating to learn about, you know, when you're first learning about chromatin is you're like, well, you just have to figure it out as you go. DNA methylation is kind of great because I think it always represses gene expression. And that's what it does. It just reduces transcription. You know, you can't transcribe this gene, the end. And then histone acetylation is also a good one because I think it always opens up chromatin. It always allows expression. Some of the other modifications of histones vary depending on the different gene, the different context like that. And so that's the why I mentioned histone methylation. And our book talks about there, and it just gets complicated and frustrating. But we're going to go through there with that, and then maybe add some more, you know, a little bit more in our learning in chapter 9, like that. So DNA methylation, histone acetylation, and then nucleosome phasing, and we'll talk about nucleosome phasing in just a little bit. Other questions? Let's see. There was another one. Hannah, did you have one? Huh?
That was actually my question.
All right. Other questions, issues people have? Okay, let's just look where we're at. So, chromatin, and that's a big part of chapter 9. So chapter 9 is gene expression by regulation transcription. So it's our original clay model videos. Then there's this chunk of it that is about the regulation of transcription. And so we're looking at examples of rate-limiting steps. And sometimes it's at the pre-initiation complex, sometimes it's in the chromatin folding, sometimes it's this arrested polymerase. So this is all fitting into that chromatin remodeling that we'll talk about today. And we're just going to finish today by starting to talk a little bit about this arrested polymerase. So that's another example. That's that thing where you go to the fair and you're getting a burger, and the person that's the last person who's actually serving it just won't let go of the plate. And they're just like, say, is that a new hat? That's really cute on you. You know, and you're just like, give me my, you know, like that, right? And it's weird that that's the rate limiting step, but it can be, right? The pickle lady's doing her job. It's this server that's just way slow. And that happens. Pre-initiation complex, great. RNA polymerase is there, starts to take off. The chromatin's unfolded. It's open, euchromatin. And the polymerase goes to about 70 nucleotides along that transcription unit, and then it just stops. And that's the rate limiting step. We need to get that arrested polymerase released and kick it, get it going again. And that's what some transcription factors do, and our example is a transcription factor we talked about called heat shock transcription factor, or HSF. And so we'll just draw a little drawing of what that transcription factor does. And it looks like this. Here's a gene, it's called a heat shock gene. And there are several heat shock genes in us. And in every animal, in every plant, maybe every eukaryotic cell has these heat shock genes, and it has a heat shock response. And the heat shock response is that HSF gets activated and binds to it and activates expression of the heat shock genes. And heat shock can do that, but what it is it's more generally a type of stress response that an individual cell makes, and it makes it when something happens that causes a lot of proteins to denature. And so the heat shock response, think of as like an EMT response, first aid response. This cell would die unless we can rescue it. And the way it does that is it quickly makes these heat shock genes, which make heat shock proteins, and the proteins, the heat shock proteins, help refold denatured proteins. And you've learned in hydrobio that a lot of times denatured proteins, they're ruined. But it turns out that in some cases you can refold them, and that's the function of these heat shock proteins. And there are probably about a dozen different heat shock genes, and they all do that. They all try to rescue the cell from imminent death, like that. So think of it as, whoa, we have to make a fast and big response. So one thing is these heat shock genes, regulatory regions, can just produce tremendous amounts of product. So remember, the regulatory region regulates where, when, and how much. And so for the heat shock regulatory region to be like, well, every single cell, when, stress, some kind of stress, and then how much, and it's like tons now. So these genes were studied a long time ago because they were such good examples of transcription regulation. That's why people really studied them as an example. And that's HSF, and what's it doing, and this is our last concept for today, and what it is doing is it is kicking that arrested polymerase in the butt and just getting it going. So the rate limiting step are these heat shock genes, and right now in our bodies, the heat shock genes all have an arrested polymerase, and they're just waiting for trouble. So it's really a sweet example because it's like when you want this gene expressed, you want it expressed as fast as possible. So it's actually a really good classic example of an arrested polymerase because it's just like, I am so ready to be expressed, but I'm not. And then heat shock gets transcription factor gets activated and go like that. And then it goes, and then it's just there, and it's just loading these polymerases as fast as it can, like that. It releases one, and the next one is actually already there, and it gets to that point, and it just releases it. So you just have a high density of polymerases on the transcription unit. Okay, we gotta stop. So we'll talk about this, and I think we'll finish up chapter 9 stuff on Thursday.